Thank you guys for getting back in here. Let's get started. I'm Brittany Parke, and the research coordinator for HCU Network America. And I'm providing with you basically a research update. I'd like to think of this as like the culmination of today's information, right? Hopefully this morning you heard from some phenomenal speakers about what is homocystinuria. We heard about what the treatment guidelines are. You heard from Dr. Kruger about what different potential treatments are out there. And Kristen gave us an amazing overview of, why research is important and ways for us to get involved. So think of my presentation as kind of like the culmination of all that information. And it's something we call like a research map. So where is research going? What do we need to do? Where are the gaps in research? So this is my family. We very rarely get family photos, so I had to stick this in my slide because I paid a lot of money for it, so I have to use it. But this is our family. I am more importantly than the research coordinator. The reason I'm here is because I'm a mom. I have two boys who were born with cblG. We lost our first son in 2011. cblG is not a newborn screened disorder. It's not picked up until symptoms start. And unfortunately, our son's symptoms were so severe, he passed away. And it was obviously really hard. And when our son Grayson was born, we had the foresight to be able to test him ahead of time. He is being treated since day one. And as you can see, he is thriving and he's doing incredible. And so this is the reason why I'm here. This is the reason why I'm part of HCU Network America, and why I love this community. Because our whole family's been really blessed with being a part of this community. So let's talk about this pathway. No one likes looking at these pathways. My husband has banned me from showing him diagrams and pathways because I show him way too many. And I'm like, does this make sense? And he's like, yeah, don't go there. So really what we're looking at is where in the pathway are some potential opportunities for treatment. So you can see at the beginning, let's back up. This is just classical homocystinuria for right now, okay? So, and then we're gonna talk about remethylation. So, is this a pointer? No. Darn it. Okay, that's okay. So at the top you see methionine. We know with classical homocystinuria patients, there is an elevation of methionine. And so that is a potential point of therapy. You could have, and we're going to talk about these individual therapies in a moment. You also have small molecule therapy that support that pathway. That could be, and currently that would be betaine, right? That current is a small molecule that supports that pathway. And then we also have therapeutics that could potentially interact directly with that enzyme. And so that would, if many of you heard next door, the Travere enzyme replacement therapy. It also would involve a chaperone therapy, which we're going to talk about what it is, as well as gene therapy or gene editing affecting that actual enzyme. So let's look at the pathway. So we know that the ultimate goal is we need better, more therapeutics. We know that there are therapies that are helping our patients live longer, but we want now to improve quality of life. And that could be eating more protein or having to take medication less often. And so that basic understanding of science and moving towards new treatments is really the goal. So when we look at the phases of the clinical trial, just like Kristen had explained to us, this is kind of what our landscape looks like right now for classical homocystinuria. We've got quite a few therapies that we're looking at that have made basic discoveries through basic research. Many of those have been funded through HCU Network America. We have many that, based on that basic discovery, have moved into some of that preclinical work. Could this be safe enough for a clinical trial? And then we do have a few that are in that clinical trial phase. Like for example, phase 3 clinical trial for Travere, as well as the possibility of an at-home monitor for homocysteine, right? So, and again, I'm gonna go through each one of these individually, but this is kind of what the outlook looks like. And remember what Kristen said, right? There is that valley of death. There's going to be some attrition, right? And we've already lost a lot, right? There's companies that go bankrupt. They don't have funding. And so we want to keep as many of these options available as possible for our patients. So starting at the top, we chaperone therapy. So I'm going to give maybe an example. Okay, so the CBS enzyme, when it's made incorrectly, it doesn't fold into the right shape. So who likes origami or paper airplane folding, right? Yeah, thank you. We like folding. It's fun, right? My son loves making paper airplanes, mostly to throw at his sister, but he likes folding paper airplanes. So you follow instructions. Those instructions tell you step by step how to fold that paper airplane. Well, what happens if you don't follow those steps correctly? It doesn't work, right? Or, you accidentally fold your paper, hamburger, not hot dog style, right? Remember that from elementary school? And it turns out funny, it doesn't fly as well. That's how I like to think about enzyme folding, right? You have your DNA communicates to the mRNA, and those instructions are passed on to create this enzyme, right? And in this instance, the CBS enzyme. And that enzyme has a function. And so when it doesn't fold correctly, it can't function properly. And so Thomas Majtan, he has done a lot of basic research in understanding what happens when those enzymes are folded incorrectly and how can we fix it? How can we rescue it, right? So if you make a paper airplane and one of the wings isn't folded properly, could you add some tape, right? This is a very simplistic way of saying it, but how can we fix this enzyme so it could function enough to give us more activity? Okay, so he developed this ability to test and look at these different enzymes. And his idea was, could we find a drug, a medication that could stabilize or help that enzyme fold correctly? So if the instructions aren't complete, if you're missing a few steps, you could then get that. The drug would then help with that binding and hold that molecule together. So that's what basically chaperone therapy is and how to fold those enzymes correctly. The same thing with a similar researcher in the UK named Thomas McCorvey. He's also been working on some chaperone therapy. So he studies the CBS enzyme in great detail. And you can kind of think of him as an engineer, right? So he looks at this broken machine and he's trying to figure out how to fix it, right? That's how I think of our researchers when they're looking at chaperone therapy. What parts are damaged? How is the machine supposed to work? What's required to repair it? And so Dr. McCorvey has been using some high-powered imaging to really create a detailed picture of what the CBS enzyme looks like and each of its individual parts and how they function. Because if you actually look under a microscope at an enzyme, it's not a pretty little blob like that. It's all folded up and intricate, and it's very detailed. So one of his major goals in his research is to find a medicine that can attach to the CBS molecule to help it work better, similar to what Thomas Majtan was doing as well. And so these are called pharmacological chaperones, and they're just tiny little helpers that help that wobbly little machine so they can do their job again. So if successful, this research could lead to new treatments and make it easier for patients to have better control of their homocysteine. And I will mention, actually, to go back a slide, Thomas Majtan, he did recently publish, I forgot to make a comment, but he recently published an article because he found medication that is used in Duchenne muscular dystrophy, and it's called givinostat, and it's a medication, and he found that it actually helps rescue and act as a chaperone for the CBS molecule. So fascinating that he actually found a molecule that works on that. Okay. So gene therapy, that's the hot topic right now, isn't it? We're all talking about ever since baby KJ, right? I don't know if you guys all know who baby KJ is. In Philadelphia, we saw that story kind of hit the news. We have heard a lot about how gene therapy can help essentially cure, the diagnosis. I say cure because it's not really a cure, right? You continually have to generally get treatment depending on what that actual therapy is. But the idea behind gene therapy is you deliver a healthy copy of the gene into someone's body, which would then create a working CBS gene, right? It's not changing your fundamental DNA. It's giving you the correct instructions and bypassing your own DNA to create that CBS enzyme. So we actually have two researchers, and they're here, which is super exciting. So you can meet them. So Dr. Warren Kruger, he's at the Fox Chase Center. He's actually been working on gene therapy for quite a while, and looking at rescuing the CBS gene. And there's also Dr. Leo Dielman. So he came all the way from Norway, from the Netherlands, to be at our conference. He's going to have a poster right after our session with Dr. George Spaeth. There's going to be a poster session right directly down the hall, and he will be over there with his research talking about also gene therapy for classical homocystinuria as well, and some of the results he's had in a mouse model. Okay, so this is actually his paper regarding the AAV gene therapy vector. And the reason I spoke up earlier, if you were in our session with Kristen, is because this is a classic example of how if a company goes under but then sells their assets and their intellectual property right, something could get shelved and we could never see it again, right? So this is a therapy that was owned by a different company. They developed this gene therapy tool, but it was then sold to another company. And the question is that company interested in Leo continuing that research or someone else taking that intellectual property right? That's kind of what we do. We go and we approach them and try to figure that out. And if they aren't, is that going to be lost to us until that intellectual property right runs out? Like, we can't access that gene therapy now because of the way that these companies are working. And so with this, So that's why it's really important for all of us to be involved in the research, because we don't want to lose this, right? And as we get involved, we can continue to have these great options. So switching gears from first chaperone therapy, gene therapy, now we're talking about oral enzyme therapy. So Petri Bio has been a part, they've been at our conferences before. They have been trying to develop and have developed, it's called bacterial derived, I can't say that, whatever that is on the screen, methionase. Yes, so you can see on this fantastic picture they provided for me, you take the bacteria or that like a probiotic or prebiotic by mouth, right? And that engineered bacteria travels into your gut, into your intestines. The bacteria reaches the gut where they live and temporarily they do their work in there. And it sucks methionine in like a vacuum. So the kind of any excess of that methionine, because right, we know that when you eat methionine, it goes into the gut. And then when it gets into the bloodstream, if you have too much, that's when you start to have elevated methionine and have symptoms that are not great for classical homocystinuria patients. So the idea is catch it before it gets in the bloodstream, before it becomes a problem. So if you were to ingest more methionine, taking that oral enzyme therapy would suck that methionine out of your gut and get rid of it before it becomes an issue. So they actually do a lot of their preclinical research with Ken McLean's lab and their mouse model in Colorado. And they sadly couldn't be with us at this conference because they are really busy, but wanted to give an update on how everything is going, that they have developed a second optimized strain, designing a third, and with each typical strain of bacteria that they're engineering, they're becoming more efficient and work better, is the idea. They're more regulatory friendly and have a plan to test their efficacy against their previous version to determine which candidate would be best to move forward. Similar to the Petri Bio, this is also another oral enzyme therapy. So Syntis Bio, I believe they're here as well if you wanted to talk to them about this possible research project and therapeutic. So they're currently in IND enabling studies. They're trying to determine proof of concept and then move this into a clinical trial setting. And so theirs is a little bit different in that it would be inactive all day removing an extended release technology to, sorry, my computer is silly, to remove the methionine from your body. So the methionine from your food enters the stomach. You can see here, and intestines. The oral enzyme pill is taken and works inside the gut before it's even absorbed, which means less methionine enters the bloodstream. And hopefully then reducing methionine levels, reducing homocysteine levels at the same time. Okay, right, and this IVD Solutions. So if you've noticed in the office across from registration, there's been some phlebotomists taking blood. You might be asking what they're doing, but they're part of IVD Solutions. So IVDS is a biotech company who are developing an at-home homocysteine monitor. It's called HCU, HCY now. And the idea would be being able to keep track of homocysteine levels at home, reducing the need for those continuous lab draws. So they are taking blood, and they could explain to you more why they're doing that to validate that that monitor as well. Because we know one of the biggest challenges is just keeping track of those homocysteine levels. If you have little ones and you understand, or even you yourself, it's not fun having to constantly go get labs. When Grayson has to go to get labs, he misses half a day of school because that's just the way it is. It takes us time to drive out there, to sit in the waiting room, to get labs drawn, to then get him lunch, and then take him back to school, and he misses half a day. And so being able to utilize something at home where he's not missing school or having to do that all the time would definitely improve quality of life. So in 2025, IVDS received an NIH CBER grant to develop this system, and they're focusing on making sure that, there we go. They're checking its accuracy. So part of the reason that they're taking blood in here now is to test the blood levels based upon current testing protocols and their home monitor to determine how accurate that protocol is. And so next is metabolic pathway modifications. So some of you might have heard Dr. Ken McLean talk next door earlier today. So he was talking about how you can use small molecules. So a small molecule would be ingested orally to support the pathway. And that would be similar to betaine that most of our patients are taking. And so he looked at how Formate acts almost as like a supercharger for betaine and in that pathway. And they are, and this is HCU funded research that we've helped fund as well. There is currently a phase 1 clinical trial in Australia, and they're looking at the lowering homocysteine through this added supercharge of Formate. In addition to that, they also discovered that adding taurine really does assist, and he can talk more about the technical stuff. I think he actually might have already left for the day, but if you ever need to get in touch with him, let me know, and you can always send him an email or give him a call. But how Formate and taurine are beneficial in cardiovascular health, in supporting bone development and growth for that. So that is his research that he's working on right now. And then last but not least, the enzyme replacement therapy. So Travere is obviously a big presence here. We're excited that they're in a phase 3 trial for pegtibatinase, which is an enzyme replacement therapy. So if you think people with HCU are missing that working CBS enzyme, so the medication that they have, the enzyme replacement therapy, doesn't fix their individual CBS enzyme. It's replacing it with a working model, right? And so if we're talking about those paper airplanes that are incorrectly folded, you're ignoring basically that and just giving them a whole new airplane, right? That can function and work properly and do what it's supposed to do. And so they are Science 37 is here, and I know that they are, if you're interested in being part of the phase 3 clinical trial, make sure you check in with them. They've got lots of information to talk about how that protocol works. And they also have a poster which talks about how the study actually is run that we'll have later today. Okay. Just want to make sure I have enough time. So now we're going to switch gears a little bit from classical homocystinuria. So those are what's on the forefront for classical homocystinuria. Now we're going to talk about remethylation disorders. And this gets a little bit more complicated because if you remember my model from the very beginning, there was one enzyme, right? Now when we talk about remethylation disorders, you see all those little green blobs. Those are all different enzymes. So each individual cobalamin disorder has each different individual enzyme. And so the hardest part about this is if a therapy works for cobalamin C, it does not necessarily mean it's going to work for cobalamin G or cobalamin F. And so really it's hard to say, could one therapy be for all remethylation disorders? Probably not, right? So if we're looking at the possibility of an enzyme replacement therapy in the future, that would have to be an enzyme replacement therapy for every single one of those individual enzymes because they are different. They have different DNA that code to different instructions that create different enzymes. And each one of those enzymes has a different function in that pathway. And so you can see, we know that hydroxycobalamin, right? High concentrations of hydroxycobalamin. I'm sure Dr. Manoli talked about that this morning if you were in her talk. Is where we need to have, that's the most important part of therapy for remethylation disorders. But there's a huge problem, and that's access, right? We can get it sometimes. I mean, look at Kim, because she's in California, and it sometimes is just not available. It's not FDA approved. There's no standardization. In order to get hydroxycobalamin, you have to go to a compounding pharmacy and have that shipped to you. And the standards are different. It could be different from state to state. Sometimes they come fresh, sometimes they come frozen. The concentrations are different. And so even though we can access hydroxycobalamin, it is not a standardized therapy for our population because we don't have an FDA approved version. And that's something that we have been working on very continuously about getting and improving access of this medication to our patients. And then you see each one of those little blue dots, that's the potential for an enzyme replacement therapy. You also have chaperone therapy. But again, that chaperone therapy, like CBS, would have to be individual to each one of those enzymes. And what is happening to that? So there's a lot of basic research that has to happen. We need to understand, how does this gene mutation change the folding of that protein, right? And that's a lot of that basic research that we have to fund and we have to find out, because in order to do that, we can then create a chaperone therapy to fix it. And then on top of that, again, small molecules. So looking at different molecules like formate or taurine, similar to classical homocystinuria, to support that pathway alongside betaine or folinic acid. Oh, I'm sorry that my text is all funny. Okay, so again, looking at this we have less options. There's less stuff going on than classical homocystinuria, right? There's not as many things on our timeline. But this makes me so happy to see. I don't know if any of you are here. Was anyone here 2 years ago when I spoke? Kim, Brandon, okay, you guys were here. There was nothing, okay? I stood up here and I was like, we are grateful. We're kind of a part of the NIH study with Chuck. We've got that going on. Yeah. But nobody else cares about us. It's just kind of sad and lonely out here. And I'm so excited that 2 years later I can present and have actual options of things that are happening within our community. And it is so exciting. There's interest and there's potential. And so that's what I'm excited to share today. So we're really looking at a lot of basic research, a lot of preclinical research. And that's because we don't really understand a lot of the in-depth modeling that's happening. How can we fix it? Because we don't know how it's broken, right? That's what an engineer does. They figure out why is it broken first, and then they can try to fix it. So one of the really cool projects that we've recently funded is out of ETH Zurich, and they are actually here. They flew in from Switzerland to be here. So it's Dr. Ferdinand van Meyen and his PhD student, Beata. They're phenomenal. They're growing mini brains. That's just how I like to. It's literally mini brains. It's really cool. They have pictures of it as well. So essentially, we know that within the remethylation pathway, the areas in the body that get affected the most and that are most severe are neurological. It's in the brain, right? We know a lot of times it could be because they have low methionine. They have developmental issues, vision issues, and seizures. And so what they did is, instead of creating an animal model, because the problem with an animal model is a lot of the gene mutations that cause cobalamin disorders are lethal. And these mouse models don't survive long enough to understand what's happening or to even try a new treatment. So they created this cerebral organoid. And so they took fibroblast skin cells from healthy adults, and they genetically engineered them with their magic, because they're incredibly smart, and have growing these mini brain models of what brain tissue would possibly look like in patients with cobalamin G and severe MTHFR. And the really great thing, there's a lot of potential here. They can screen for new drugs. Are there ways that improve? Like, is there a drug or is there a current medication on the market, right? We talked about how givinostat for Duchenne muscular dystrophy could be a treatment for classical homocystinuria. It's possible, right? There could be drugs out there already on the market. So they can screen current medications and current therapies that could possibly aid in the issues that occur in the brain. And so their model, this is going to be an ongoing, they're very passionate about this. This will be an ongoing research project that's going to be taking place, and I'm excited. I think great things are going to come from this. There's also Dr. Jen Sloan. She's at the NIH, and she has a poster here as well. She's also looking at and has a mouse model for gene therapy. And it's pretty cool. She's got some information on her poster, so make sure you stop by and ask her questions if you have it. But the NIH researchers have created the first viable mouse model for cobalamin C, and they've used them to study the disease and then test gene therapy. So these mice show many of the same biochemical problems that we see in our children with cobalamin C, including high homocysteine and high methylmalonic acid levels. And then in these preclinical studies and in information that she's passed on to us, she has seen that the AAV gene therapy was able to rescue newborn mice that would otherwise have had severe phenotype and not survived. They improve their survivability. It corrects their important metabolic abnormalities and shows promise for preventing complications related to cobalamin C. So check out her poster, and we'll keep an eye on this research as it continues. And then we have Dr. Brian Gilfix. So he's in Canada. He unfortunately couldn't be with us. There was, I guess, an airplane ran off the runway in Canada, and they shut the airport down. So he couldn't make it, but he presented earlier this morning about aminoglycosides. So they're essentially antibiotics engineered, and they're specific to stop codons. So when that stop codon occurs, these antibiotics, aminoglycosides, allows for the read-through, allows the full expression of that gene. And so that he presented on that earlier today, on how that works. And this is his paper. And one of the really cool things, in addition to him looking at this specific new therapeutic, he's been also finding, he's also been looking at, in cobalamin G specifically, understanding complementation analysis. So in addition to finding new genes that could cause the disorder, because one of the biggest problems is if you have, let's say, biochemically, you have high homocysteine and methylmalonic acidemia, and they do gene therapy, they might come back with a variant of unknown significance, which means we have no idea whether or not that gene causes the problem. It could, but we don't really know. And so what he's trying to do is figure that out. He wants to try to determine, does this gene cause the disease, or does it not cause the disease? And so in his recent article, he discovered 24 new variants. And so that just improves our understanding of the disease and how it can affect our patients. It also helps with diagnosis, right? So if we now know that 24 more genes can cause the disorder, when patients get gene therapy, the likelihood of them getting a VUS decreases because now we know a little bit more about the gene. Chaperone screening. So this is taking place in Italy, where Dr. Sylvia is looking at possible ways for the chaperones to help in the folding of specifically the cblC enzyme. And this is her paper that she published. I don't have a lot of time, so I'm kind of flying through these last ones, but I want to make sure you guys have time for questions. And then this is a brand new article I'm really excited about. Right before I finished my presentation, there was a publication that came out of France, and they are looking at, it's a different type of molecule, but a CERT 2104, which helps with neurological symptoms of cobalamin G. And so we know that cblG can cause severe early life brain metabolism and neurodevelopmental disruptions. And a lot of that has to do with, and may not always be relieved with the standard B12 supplementation. And so they're looking at this particular CERT 2104 has been in a phase 2 trial for safety. And looking at whether or not that can rescue and support and restore metabolism in neurological function for cblG. And then, of course, the at-home monitors for everybody, right? Because we all have high homocysteine, so it's not just for classical homocystinuria. It's for our cobalamin and remethylation families as well, which is exciting. And the formate and taurine, also looking at the possibility of how that might affect not just CBS, but also the cobalamin disorders. And this is a publication, I think most everyone probably, I'm hoping everyone, if you didn't watch or weren't in the room when Dr. Chapman and Dr. Manoli gave their talk, the importance of guidelines. So it's super exciting that EHOD just published our new guidelines for cobalamin disorder. It helps us understand and get all on the same page, right? So many times we have patients come to us and families that say their doctor doesn't understand and doesn't know how to treat this disorder. Now you have a paper, you take it to them, and it gives them more information. And this is published research. Sometimes they don't believe us because we're just the parents, right? But if they have published research, that speaks to them, okay? So global grants program, I'm gonna go over this super quick. We have been able to give 10 grants through HCU Network America, and currently are evaluating to give 2 more. And so this is kind of the timeline where we started in 2018 with Dr. Ken McLean, looking at the trying to discover a new molecule, which is what he then discovered with formate and taurine. Dr. Tomas with looking at the chaperone therapy, as well as Thomas McCorvey, looking at newborn screening in remethylation disorders and cobalamin C disease in Italy. And then most recently in Switzerland with the cerebral organoids, Dr. Gilfix with the aminoglycosides, and it just keeps building. And the exciting part is I stood up here 2 years ago and I said, I feel like I've spent the last couple of years like trying to form a snowball, right? And I was like, I feel like the snowball is finally forming and I can let it go. And as it goes down the hill, it's going to gain momentum and it's going to just keep picking up speed and it's going to keep growing bigger and bigger and bigger. And I feel like we're at the point where that snowball is starting to really tumble down the hill and it's gaining speed. It's gaining momentum. We're being heard. People see us. We're out at conferences all the time trying to advocate for our community and really helping people like see that we're here, that we want new treatments, that what we have is not good enough. So we need them to start researching it. So next steps, this is where I need all of you, okay? We can't do this without you. Those 10 grants I was talking about are because of our community. It's because you fundraise, it's because you actively are participating in our programming. And so we do have our new call for grants. We plan on this year to issue 2 more grants, one for classical homocystinuria, one for cobalamin G. We have received 6 expressions of interest. That means 6 researchers are interested in that money, which is exciting. And then we anticipate and plan to fund that later this year. I will also put a quick plug in. Being a part of our organization and not just these active research, the reason researchers are getting excited is because they hear your voice. And they hear your voice when our participants are involved in RareX, right? It might feel like you're just putting your data in and it's going out into the interspace and nobody's using it. People are using it. We're using it all the time. We've gone to 4 conferences. I'm headed to Finland later this year to present 2 posters on RareX data, and it's super duper exciting. And the more data we have and the more people are seeing it, that snowball is going to grow, the momentum is going to gain speed, and more people are going to be interested because, just like Kristen said, one of the reasons why these new drugs or therapies fail is because there isn't patient community involvement. We don't want to be that community, right? We want to be the one that helps and boosts up those therapeutics. We don't want it to fail because we weren't there to pick it up. And so being a part of RareX, we help build natural history studies. We're participating in clinical research and being a part of it. We can't be the ones on the sidelines saying someone else will do it because there is no one else. We're way too small for that. We're the ones that have to do it. And there's great potential out there. All of these projects are ongoing. We have passionate, amazing researchers that are interested in our community. And I just, if you're interested in RareX, if you're interested in being a part of the research committee, please come find me this weekend. Please email me. I am not a researcher, okay? My background is social work. But I've learned, and you can learn too. Anyone can, and I'll help you through it. If you're interested in it and you want to be a part of it and you're excited about research and you want to learn more, we can learn together because there's many, many calls I'm on with the researchers where they'll say something and I'll have to say, whoa, back up. Start like square one, help me understand this better. And they're incredible. No one's ever made me feel like I don't understand. They take the time to help me, right? That's what they're here for, and they care about our community. So my hope is in 2 years, when they make me get back up here again, I have even more projects to share. And these projects that we do have been progressing, and we've got exciting new therapeutics and options for our families, for ourselves, and that we continue to understand these disorders because that's what we're here for, right? And that's what we really want for our families. So thank you. Join the Race for Research, guys, because it's going to be a ton of fun raising money for specifically research. And if you have, again, any questions about research, we do have a few minutes until we need to join the other group. If you have any questions, I can take questions for a few minutes. But thank you. I appreciate all of you. Did anyone have any questions? No? Okay.
Give Brittany a hand.
Oh, gosh.
She has done an incredible
Thanks, Kim. And I really do mean it. Like, if I can do this, so can you, right? Like, I took Biology 101, you guys. I am not a scientist. But when you have a passion and when you have a will for something and when you know that you need to do it, it'll be there. And so if you want to join us, you want to be a part of research, please come find me, email me. We'd love to have you a part of it.